Hello students, welcome back to the biology class. Hope all of you are fine. Today I'd like to tell you something about success. What is success? Success is not an accident. It's your perseverance, it's your study, it's your learning, and moreover, if you want to be a successful person in the life, you have to love what you are doing. So students, today we are on the chapter 11. The title of the chapter is Reproduction in Organism. After finishing the lecture, students, you will be able to learn something about the Megasporogenesis. So, today our topic is about Megasporogenesis. What is Megasporogenesis? Megaspore means female gametophyte and genesis means the development. So, the development of megaspore is known as the Megasporogenesis, and megaspores are also known as female gametophyte, ovule, embryo sex, right? Okay. Students, whenever we are going to study about the female gametophyte, we have to know a basic concept about the different parts of the flower. This is a complete flower. You know, this is the stalk, this is the thalamus, and this one is the sepal. In singular, we will call it as sepal. This is petal, this is stamen, and this one is carpel or pistil. And collectively, the sepals are known as the calyx, and petals are collectively known as corolla. This one, the male reproductive structures. These are known as the androecium, and this one is the female reproductive structure. This is known as gynoecium. This one is the gynoecium. It consists of stigma, style, and ovary. Inside the ovary, one or numerous ovules are produced. Today, we will focus about this ovule, right? This is the structure of a typical anatropous ovule. In angiosperm, you will find this type of ovule. This one is the stalk, this one is the body part. Okay. Whenever you are drawing the female gametophyte, any structure of female gametophyte, you have to specify the micropylar end and the chalazal end. What is anatropous ovule? Anatropous ovule is the ovule in which this portion, what is this portion? The body part. The ovule is inverted. The anatropous ovule is that type of or this type of ovule in which the micropyle is or ovule is just bent down to this micropylar structure. Is the whole ovule is inverted and the micropylar side is bent down to the funicle. This type of ovule is known as the anatropous ovule, and you will find this type of ovule in the angiosperm, right? This is an anatropous ovule. This one is the micropylar end, this one is the chalazal end, this portion is the body part, this is the stalk part. Stalk is known as the funicle, this is micropyle. It has two integument, outer integument and inner integument. Okay, students? Okay, now we will draw a typical ovule and we will learn how the female gametophyte is produced, right? Student, this is a typical anatropous ovule and we are drawing the ovule, right? What is this? This is a typical anatropous ovule. This is an upside down ovule. The micropylar end is towards the funicle. This is the outer integument. You can draw with me. Integument. This one is inner integument. Right? This is micropyle. This is Challenger. And the central portion is known as the nuclear cells. There are numerous nuclear cells. This is the stalk funicle. Numerous nuclear cells. And today we will focus on the lower nuclear cells. Okay, students, the lower nuclear cells. And in your textbook about the origin of these nuclear cells, there are no ideas and no details about the lower nuclear cells, and we will call it as the megaspore mother cell. Megaspore mother cell, and in short we will call it as MMC, megaspore mother cell. And about the origin, I would like to tell you something about this origin. This cell derives from the archisporial cell. This cell derives from the archisporial cell. I am telling this to just satisfy your curiosity. You should know about the origin of this MMC, that means the megaspore mother cell. It derives from the archisporial cell. What archisporial cells? The archisporial cells, they are derived from the ovary wall. Ovary wall. They are hypodermal in origin. What does it mean? Hypodermal in origin means they are derived from the ovary wall. So, archisporial cells first divide and give rise to two types of cells, and the division is archisporial cell. This division is a type of parietal division, and this is denoted like this. After this parietal division, we get two types of cells, outer and inner. Outer cell and inner cell, right? And these two types of cells then undergo another type of division, and after that division we get what? Primary parietal cell, and from this primary sporogenous cell, and from this type of cell, that means primary parietal cell, further will divide or not divide depending on the nature of this type of cells. May divide or may not divide, but primary sporogenous cell will further not divide, and they will give rise to the megaspore mother cell. We are learning this to satisfy our curiosity about the origin of the megaspore mother cell. This is the megaspore mother cell. Okay, students? We'll talk about this, about the megaspore mother cell. What happens in the megaspore mother cell? One cell near the micropylar end enlarges, and this gives rise to different types of incidents. So let's see. This is the megaspore mother cell. It is diploid in nature. That means 2N. I am writing in short, megaspore mother cell. This is 2N. That means diploid in nature, and it undergoes a meiotic division. Rather, we can say reductional division. And according to the law of meiosis, all of you know that from one cell we get 4 types of cells. From one cell, 4 cells. So 1, 2, 3 and 4. And what will be the chromosome number? It will be N. This is N, N, N and also N. And these are known as linear tetrad. Linear tetrad. Again, from the megaspore mother cell we get 4 megaspores. They are known as linear tetrad, and we are getting this linear tetrad through the meiosis or meiotic or reductional division. Clear? After that, 3 cells from upper one or 3 cells from lower one may degenerate. That means upper 3 from upper side may degenerate or lower 3 may degenerate. Whatever it is, we are getting only one cell, only one survival cell. Suppose the upper 3 are degenerating in this case. 1, 2, 3 and 4. And upper 3 are degenerating. So writing upper 3 or lower 3 degenerates. What happens then? We are getting only one survival cell. Only one survival cell. That means one megaspore survives. One megaspore survives. And this survival one will, so we should draw how these are degenerates. Let's draw this. 1, 2 and 3. These are degenerating megaspores, and survival one is this. One megaspore mother cell survives. After some time it undergoes mitotic division. Karyotypes happens first. And in first mitotic division, first mitosis. What happens in mitosis? Can you say? Yes, I know. You can say. In case of mitosis, from one cell we get 2 cells. That means we are getting like this structure, 1 and 2. Right? From one cell, from first mitosis, we are getting 2 cell structure. Write down 2 cell structure. And after that, what happens? Can you see, students? What happens? So I should write some upside. I'm writing linear tetrad. From 2 cells, 2 cell structure, we are getting 4 cell structure. Why? Because now second mitosis is happening. So 1, 2, 3 and 4. We are getting 4 cell structure. Write down 4 cell structure. And after that, the third mitosis will take place. And we will get what? From 4, 8 cell, 1, 2, 3, 4. 1, 2, 3 and 4. These are the nucleus. Okay, easy? First, meiosis. So, from 1 cell, 4 cell. 3 cells are degenerated and we are getting only 1 megaspore, and this megaspore will act as functional megaspore. Functional megaspore. This functional megaspore will undergo first mitosis, and according to the law of mitosis, from 1 cell we get 2 cell. So from the first functional megaspore we are getting 2 cell structure. After that, second mitosis we are getting 4 cell structure. After that, third mitosis and we are getting 4 cell structure. Now we will talk about the arrangement of this structure. Right? I am erasing. Okay? I'll give the drawing later. Okay? And about the arrangement of the 8 cell structure, I am drawing quite larger. Can you see? In each pole there are 4 cells, right? 1, 2, 3, 4, 1, 2, 3, 4 in each pole. One pole and another pole. 1, 2 and 3. What? There are 4. I am writing 3. I am drawing 3. Yes. I am giving you the reason. Have a look. 1, 2 and 3. 1, 2 and 3. And 1 cell from each pole will migrate toward the center. Right? 1 cell from this pole and another cell from this pole. So the number is fixed. 1, 2, 3, 4, 5, 6, 7, 8. 1, 2, 3, 4, 5, 6, 7, 8. No change in number but change in the arrangement. What happens now? Wanna know? Yes. Now we should give them some name. What will we give them name? These 3 cells are known as the antipodal cell. What? Antipodal cells. Antipodal cells. I'm writing antipodal cells. Right? And in the center there are 2 cells. What happens in the 2 cells? Happens something like their cytoplasm come together but not nucleus. So this look, after some time it looks like. That means cytoplasm fuses but not nucleus. Mind it. Cytoplasm fuses, not nucleus. And this is known as secondary diploid nucleus or polar nuclei. Whatever you want you can write. Polar nuclei or secondary diploid nuclei. And these 3 cells, about these 3 cells, and one thing I should mention you about, whenever you are drawing, you should mention the specific things. That means this is the chalazal end, this is the micropylar end, this is chalazal, micropylar, chalazal, micropylar, chalazal and micropylar. What does it denote? It means that this portion is chalazal and this portion is micropylar. Okay. Now, this is secondary diploid nuclei, and this one and also this one, both sides. These 2 cells are known as synergids. Synergids. Okay. And the central cell, the central cell is known as the egg. Choose the micropylar end. The central larger cell is known as the egg cell. Right? So these 2 are synergids. The central larger cell is known as the egg, and this cell is known as the secondary diploid nucleus, and these 3 cells are collectively known as the antipodal cell. Now about the function of the antipodal cell. What are the functions of the antipodal cells? The antipodal cells provide nourishment to the developing secondary diploid nuclei. They provide nourishment. Clear? Secondary diploid nucleus. What is its function? Secondary diploid nuclei take part in the second fertilization. In this case, 2 fertilization will happen, first and second. So, secondary diploid nuclei take part in second fertilization. And what is the function? Egg take part in first fertilization, okay, and secondary diploid nucleus after some time will give rise the endosperm, and the egg will behave as the embryo. And what are the functions of the synergids? In the synergids, there are some filiform or finger-like structures. This filiform or finger-like structures are known as the filiform apparatus. Okay, this filiform apparatus attract the pollen for fertilization. Got it? This filiform, the finger-like grain, I have written, I have drawn. This is the synergids. What happens to the synergids? There are some finger-like structures, and these finger-like structures attract the pollen tube for fertilization. And 1, 2, 3, these cells together are known as the egg apparatus. Egg apparatus. So you have got a clear concept about the megaspore and megasporogenesis. Right, students? And the entire structure is known as the polygonum type, and this is a monosporic type of embryo sac. What does it mean, that monosporic type? Monosporic means we'll get the answer from our drawing. Students, have a look about this structure. Can you see? Yes? 1, 2 and 3. These structures. Should I draw again? Okay. 1, 2 and 3 and 4. These 3 megaspores have degenerated, and we have got only one functional megaspore. In this case, 3 cells out of 4 cells, 3 cells have degenerated, and the female gametophyte has originated from only one functional megaspore. Got it? Out of 4 cells, 3 have degenerated, and we are getting only one functional megaspore. These type of embryo sac are known as the monosporic type, mono, 1. So, 3 out of 4, 3 megaspores degenerated and 1 megaspore survives. Three megaspores degenerated, only 1 survives. So monosporic. And out of 4 megaspores, when in some angiosperms, 2 megaspores degenerated and 2 survives, and this is known as disporic type. Out of 4 megaspores, one megaspore degenerates and 3 survive. These type of embryo sacs are known as the trisporic. And none of the megaspore degenerate and 4 survive. This type of megaspores or embryo sacs are known as the tetrasporic. Right, students? So, about the characters of our embryo sac or female gametophyte, in short, we can write 3 characters. Should I rub this? I am rubbing this, and I should write the 3 characters. Number 1, our female gametophyte is polygonum type. Polygonum type. Number 2, it is monosporic type. Monosporic type. And number 3, who can say the number 3? I will say. For that, I should count the number of cells. 1, 2, 3, 4, 5. 1, 2, 3, 4, 5, 6, 7. 7 celled. 7 celled. And number of nucleus? 1, 2, 3, 4, 5, 6, 7 and 8. 7 celled, 8 nucleated structure. So, in brief, we can say that our female gametophyte is polygonum type, monosporic type, and 7 cell, 8 nucleated structure. And what about the number of chromosomes? Antipodal cells are n in number, and the secondary diploid nucleus 2n, and the synergids and eggs all are n. Students, this is the megasporogenesis in short. You can follow this flowchart. That means this is the function, this was the megaspore mother cell. 2n, diploid in nature, it undergoes meiosis and produces 4 haploid nucleus or megaspores. These are 1, 2, 3, 4. These are known as the linear tetrad. And these upper 3 or lower 3 will degenerate and produces, when upper 3 degenerate, produce 1 functional megaspore. After that, this functional megaspore undergo first mitosis and produces 2-stage cell, that means 2 cell, and second meiosis, 4-cell stage, third meiosis, 8-cell stage, and in this 8-cell cell, 1 cell from each pore comes towards the or migrates towards the center, and this is the structure. And this, I have told you earlier that these 3 are known as antipodal cell. The whole structure, entire structure is known as the embryo sac, center, secondary diploid nucleus. The larger one is egg, and these 2 cells are known as the synergids. This is 8 nucleated and 7 cell structure. You can follow the flowchart for your understanding. Okay, students? Students, I think that I should finish my lecture here. If you face any type of problem understanding the megasporogenesis, you should watch the video again and again. Till then, your Papiya Miss is saying goodbye to all of you. Take care, keep watching, see you later.